Hello everybody, and welcome to our October 2016 Discovery Series presentation, which we are recording here in our home studio at CFRI. I'm Siri Vathan, and I'm the Programs and Outreach Manager at CFRI, and I'm very pleased to welcome everyone here to this event. We have a very special program for you here tonight, but before getting started, I want to thank Vertex Pharmaceuticals, Genentech, and Chiesi USA for generously sponsoring the Discovery Series. And I also want to thank Gilead Sciences and Novartis for their additional support. As we stress at every Discovery Series, we urge you to work with your medical care team and to please note that no information presented tonight is intended for a patient's diagnosis or treatment. Tonight's program is being produced by your CFRI Live team, Scott Wakefield, David Suhu, and Mary Convento. So let's get started. The title of tonight's presentation is What is New in 2016, where we explore developments along the CF therapeutic pipeline. And we are so honored to have Dr. Dennis Nielson with us here this evening. Dr. Nielson is the director of the CF Center at UCSF Benioff Children's Hospitals. He is a nationally renowned CF clinician, CF researcher, and also a dedicated CFRI volunteer. It is no wonder that CFRI honored him with the CF Champion Award in 2015. Please help me welcome Dr. Dennis Nielson.
Thank you for that great introduction. I hope I can live up to this, or I'll do my best. Okay, so I didn't put a disclosure slide in here, but I will tell you that I have received support from a little bit from Gilead, and we're in the beginning of trying to do a study, what I think is a very important study with Vertex Pharmaceuticals. And I'll try not to say anything that shows any particular bias. Okay, so some of this will just be a review, and I don't know how many people are new to this. Most people, I'm sure, know all about cystic fibrosis, what it is, what it does. But basically what it is it's a channel in the membrane of cells, which you see depicted here on the left. There are these pores that develop that really drive an awful lot of what keeps us alive. Ion movement is what makes the current occur in the heart, that keeps the heart beating, that really triggers or allows your thoughts, your brain to work. But in a very much simpler sense, these ion channels exist in virtually all epithelial cells in the body, including the lungs, including the pancreas. And it's a defect or an absence of these channels that causes CF. And what happens with the absence, or if there are channels there, they don't function very well, is that you have decreased secretion of fluid from glands in the airway. And humans are special in the regard that we have these glands in our airways that many animals do not. So we're a little different in that regard. We seem to depend on these glands for secretion of fluid, water, and salt to liquefy the secretions in the lung. This may also be a defect in the epithelium on the surface, so these lining cells also may secrete some fluid. And one of the things that we know is that there's also absorption of salt and water, and that there's a balance between these two processes that is carefully controlled by the cells, and that really are responsible for making lung secretions relatively thin, easy to clear. And when this process is imbalanced, then problems follow. So one of the things that happens in CF is that there is unopposed absorption of salt and water from the airway surface. so that what you end up with is that this lining liquid layer is too thin. And this is an important thing because there are little projections from the cells in the lung that are called cilia. And they constantly beat and sweep, and they sweep mucus up the airway. They sweep, in that mucus there may be bacteria or particles that are trapped. And this is a very important mechanism for keeping your lung clean. And so we think this is an important part of what happens in cystic fibrosis. It affects the airways, the lungs, the sinuses, which is why if you look at sinus films in CF patients, by the time kids are 9 or 10 years old, they're virtually all abnormal. They all have a type of chronic sinusitis. It's why the reproductive tract in males doesn't develop properly, because there are also cilia there that are an important part of that body system. In any case, this results in a very slow or even just stoppage of the clearance of these particles. There's another important thing, though, that happens and that is that this channel, the CF channel, which transports negative ions, chloride is the most important and most abundant one that transports, but it also does move bicarbonate. And bicarbonate is a negatively charged ion that is very important in balancing the acidity levels in all your body fluids, which include the blood, the interstitial fluids, and also the airway fluid. And it turns out that that bicarbonate problem, and also was discovered actually this year, part of the update is that there is a hydrogen ion, an acid pump, that seems to work in concert with the CF channel that has been described in the last year, and that the absence of cystic fibrosis has an impact, or absence of the channel, affects the function of this acid secreting pump, and that it secretes too much, so that there's a lack of control. The importance of this is that there are molecules that we all secrete. The epithelial cells make them. They're antimicrobial. So, the human body and presumably cells in general, all creatures learned long ago in their development to actually make antimicrobials. So it wasn't, penicillin wasn't the first antimicrobial ever made. It was actually made by cells of all types as a defense mechanism. So this is also an important part of our body's ability to keep the lung clean. And having too much acid affects how these molecules function, so that they're not able to kill the type of bacteria that people with CF get in their lungs. So a consequence of this defective salt and water movement is that you have all kinds of problems in many organs in the body. Anywhere where there's an epithelium that's important, you may or may not have problems. I mean, one of the interesting things is that we know that the CF protein, the CF gene, is expressed in the kidney, and yet kidneys seem to work pretty well, and they're really not affected by this problem. But if you look at other organs, like the intestinal tract, this abnormality has a huge impact on how well the intestines work, and the bacterial environment in the intestine is abnormal in CF patients because of some of these abnormalities, including not only the lack of mucus, but also this acidity, the pH problem. It affects the pancreas, we know, because the ducts in the pancreas are actually, in many cases, in the majority of cases, abnormal at the time a child is born, and they're unable to secrete pancreatic enzymes because those ducts didn't develop properly because of a lack of fluid movement in the developing fetus. The liver is affected, although it's a relatively small group that develop liver failure. But as I said before, everybody who has CF, virtually everyone, gets chronic sinusitis. Some people get polyps, which are a result of that chronic inflammation. But the thing that really threatens the health and the life of CF patients are the frequent and chronic lung infections that occur as a result of these abnormalities in salt and fluid movement, and probably this problem with acidity in the secretions. So, long ago we learned how to do what I call downstream treatments. In other words, they're downstream from the gene, they're downstream from the product, they're downstream from the cell. We're just looking at sort of the whole organism, the whole body, and trying to compensate for the problems that CF causes. So I'm not going to do this in any chronological order, but we're just going to go through here, and one of the things that we learned several decades ago was that in CF, nutritional management is incredibly important. It affects not only growth, sexual development, but it also affects lung health to a great degree. And that if children with CF grow well, they tend to do better in almost every way. So that we have developed pancreatic enzymes, nutritional supplements. Anyone who's been around CF knows that we do things like gastrostomy, feeding tubes, sometimes if we can't keep up with the needs. All kinds of interventions have been designed. And what's really coming through the pipeline in CF is summarized here, and this is just taken directly from the CF Foundation website. And if you go there, you'll see this, the Aqua Dex, which are a water-solubilized form of fat-soluble vitamins, so that they're more easily absorbed. And so the fat-soluble vitamins are A, D, E, and K. Makes a nice little word that really doesn't mean anything as far as I know, although it could be a superhero that Stan Lee named some time ago, and I just don't know about it because there are hundreds of those. But as far as I know, ADEX doesn't stand for anything other than A, D, E, and K. And those are your fat-soluble vitamins. So that's something that has gone from preclinical, phase 1 trials, phase 2, phase 3, and patients are now benefiting from that. Pancreatic enzyme products, this goes back actually many years, and for many, many years and decades, pancreatic enzymes were treated as a food supplement. So they were all based on pancreatic enzymes that were harvested from pigs in a giant slaughterhouse somewhere in the country. And they purify the enzymes, and these were put in capsules, and the capsules then were put into granules, and we have these fancy coatings that are pH sensitive, and all of these things that have developed over the years. But it was only relatively recently, because of a problem with insurance companies trying to force patients to take generic enzymes, that the FDA went back and forced the pancreatic enzyme manufacturers to go through a testing protocol. So these now are actually approved by FDA, they're tested so that you know what you're getting is what's on the label. Lipoprotease is an interesting product in that this is a non-animal based enzyme. And this is important because there may be rare people who are actually allergic to pig enzymes, but pigs are close enough to people that doesn't happen very often. But there certainly are religious problems that come from using a pig enzyme, and this applies to obviously to people who are Jewish or who are Muslim, and this is, people get dispensation to take these. Patients have gotten dispensation, but it certainly alleviates that problem if you have a product that works well and that doesn't present them with that paradox that they feel uncomfortable using a product like this. And it's also thought that the lipoprotease may have some other advantages. At this point, I can't say that we really know that that's true. You can see it's into phase 3 testing, so presumably we'll see something from this within the next year or two. And then glutathione is an interesting product. It's actually something you can take by mouth that has anti-inflammatory properties, and it's something that is natural. And it was actually suggested quite some decades ago as a possible help in cystic fibrosis, but there was never any really good testing of it until just the last few years. And there have been good studies done in Europe that show that this product can be absorbed, and we're now trying to figure out whether it really works in terms of actually affecting clinical course of CF. So we're in phase 2 there, and that's the phase where, as you discover and test a product, you're trying to figure out whether it's effective. So phase 1 is safety, phase 2 is safety and efficacy, phase 3 are the large clinical trials that still look at safety and efficacy, but they really try to, that's the step that you have before products are approved by FDA and released to the public for sale. So glutathione is in phase 2 now. So another of the classic downstream treatments are things that we do to try to restore airway surface liquid volume or to normalize mucus in the airway. We know that both of those things are abnormal in CF, that the airway surface liquid volume is low because of the ion transport problem. And the mucus also is very abnormal. It's dehydrated, it doesn't release properly, tends to be extra chunky in CF, and over time it starts to clog things up because it doesn't transport properly because of the defective mucociliary clearance. So over the years we've had things, we know Pulmozyme was released back in the 1990s, and more recently, I forget how many years now, hypertonic saline, which is just concentrated salt solutions. There were actually good controlled trials that showed that this helped pull water into the airway and had a very significant clinical benefit. Very comparable to Pulmozyme, and has the advantage that it doesn't really depend on whether or not there's infection. It doesn't care whether there are bacteria there or not. It's all physics. It just is a very concentrated salt solution. The airway lining is very permeable to water, and it just pulls the water in to equalize the salt concentration on both sides of that membrane. So it's actually pulling water out of cells, out of interstitial spaces, getting into the airway, and hopefully floating some of those little mucus boats that are stuck there on the reef and help them move downstream. Inhaled mannitol is an interesting thing. This is in phase 3 trials, so we may see this on the market soon. Mannitol is a non-absorbable, non-digestible sugar. And so it also, by being very concentrated, will pull water in as the fluid on either side of a cell membrane tries to develop what's called an equal osmotic pressure. So cells don't like osmotic pressure gradients. They don't like to have lots of salt on one side and not much salt on the other. So mannitol is not a charged particle, it's not an ion, it's just this sugar. And it pulls the water in, and it tastes a little sweet, and it's non-toxic, and it would be much nicer to inhale than these hypertonic saline solutions, which are very, very salty. They're 8 times more concentrated than normal body fluids. Oligo-G is a product that just now is going into phase 2. I won't spend a lot of time on it. It's a type of anti-inflammatory, and VX371. So this and that product now belongs to the Vertex Corporation. It formerly had another name before they bought it, but this is again another type of. This actually is an ENaC inhibitor. And then there are a couple of other companies that have gotten into the game here, and you can see these new products that were not on the list last year. This ORP 100, and the Spirax company has a contract with the CF Foundation to develop some products. Okay, another thing we've learned over the years is one part of the problem in CF is that the lungs are full of pus, they're inflamed. If you look at the lining tissue or the airway lining in a CF lung, it's red. If you touch the cells, the lining, it breaks down, it tends to bleed a little bit. It's very, very tender and inflamed. And so decreasing inflammation, we found, is a very useful thing to do. So over the years, we've done a number of things. In the old days, there were some steroid trials. We saw actually clinical benefit, but there was also a lot of harm that came with that because of the side effects of steroids. So currently steroids, even inhaled steroids, are not recommended in cystic fibrosis. If you have asthma, inhaled steroids help, but if you just have CF and not asthma, the foundation actually has a guideline that recommends against using inhaled steroids. So the rest of these molecules are different kinds of anti-inflammatories that either block inflammatory molecules in the body, or they stimulate anti-inflammatory molecules. So I'm not going to go through these in detail, because you can see the ones that are best developed are at phase 2, and the only one that actually is available to patients right now is high-dose ibuprofen. That is a therapy that, although it's been around for quite a long time, it's not been very popular, because ibuprofen in high doses given for periods of time, at least in most people's hands, tends to cause some unpleasant side effects. And so it seems to not have gained a lot of popularity in the CF community. So there are 5 anti-inflammatory molecules that are actually in testing right now, so that next year we may see 1 or more of these in phase 3 trials. So there's always lots of new stuff coming. All right, so after inflammation, fighting infection, this is something that goes back to the very beginning of CF care. Some of the first papers that were written about treating CF rather than expecting that all the kids would just die. One of the early papers was one in which Schwachman and colleagues invented a scale called the Schwachman score, and they actually used it to show that patients got better if you treated them with antibiotics, and if you did things to try to clear the mucus from their chest, and if you put them on different kinds of diets. And so this was actually, for a long time, we used the so-called Schwachman score as an indicator of severity of disease, but it was actually originally intended as a marker of response to therapy. So, fighting infection. So that's been something from the very beginning, and we know that a lot of antibiotics came about long after CF was discovered. Good anti-staphylococcal antibiotics are not all that old, but the foundation has really been involved in the antibiotic story here for the last 20 to 30 years. And if we look at things that the foundation has actually helped develop, these include 4 antibiotic types that are now in the patient arena that we use as tools routinely in CF care. That includes azithromycin, which is a macrolide antibiotic. It's related to erythromycin, but it's been shown to inhibit the formation of what people call the biofilm. So it's this slimy covering, and if you get electron microscopy cross-section, it looks like little Smurf villages with little mushrooms that the bacteria are hiding in. So this inhibits the ability of the bacteria to form these little mushroom-like villages that they use to protect themselves from things like antibiotics. So it helps the body fight the infection, it helps other antibiotics fight the infection, and it's been a useful therapy. Aztreonam, or Caistin is the brand name. Aztreonam is an antibiotic that was actually first studied as an IV antibiotic back in the 1980s, and what we discovered was that it worked pretty well against Pseudomonas the first time you used it, and then it was one of those antibiotics where the bacteria became resistant to it very quickly. And so in general now, we don't use IV Aztreonam, but it turns out that if you take the molecule and you change it a little bit and make it so that it's not absorbed and it doesn't move from an airway into body tissues, that it actually works pretty well when you get it to high concentration. So Aztreonam inhaled actually works quite well at suppressing Pseudomonas and other resistant bacteria. So it's been a very useful product. Inhaled tobramycin, which is now there's a generic form, and Toby was the brand that we used for many years. That was in a nebulizer solution. Foundation had a huge part in developing that. That was one of the things, along with Pulmozyme, that we got back in the mid-1990s. And so that's there. Inhaled tobramycin powder, the Toby Podhaler, which makes it a lot faster to inhale. All these things that we ask people to do take so much time that people are trying to find ways of making therapies for CF work, that not only maintain the benefit of the things that we've used in the past, but that also save time, because we keep piling on all these stepwise therapies, and it becomes gets to a point where it becomes quite burdensome. Inhaled levofloxacin. Levofloxacin is another antibiotic that works against what are called gram-negative bacteria. So this is in phase 3 trials, and I'm sure we'll see whether or not it looks comparable to the benefit of aztreonam and tobramycin. That will be coming out in this next year. Inhaled liposomal amikacin. Amikacin is another drug that's in the same family as tobramycin, but it's been packaged in these little fat globules, which has some advantages. Those fat globules shed cells, they fuse with the cell membrane, release the amikacin inside the cell, so you don't have to give it. This is normally an intravenous drug, and when you inhale it, it doesn't get very well absorbed, but here it actually gets into certain cells, and it gets into the biofilm, and it has some advantages in terms of being able to kill the bacteria. Then there are things that are in phase 3, like fosfomycin, tobramycin inhalation solution that are in trials, may have some advantages. We have to wait and see what the data show. Vancomycin inhalation, this is something that we use for a thing called MRSA, multiply or methicillin-resistant Staph aureus, and this has been an organism, a bacterium, that's become a big problem for us. And vancomycin is one of our mainstays, and this is a way of, rather than giving vancomycin IV, which has a lot of toxicity and a lot of risk, putting it into a powder that you can inhale that will then fight the MRSA infection without creating risk for side effects. Gallium is an interesting thing. Gallium is a metal, and it's actually approved as an IV for IV treatments for things unrelated to CF, and it has some antibacterial effects. So it's being looked at in CF. It's now in phase 2. So it passed through the toxicity studies, and now we're looking for efficacy. And then nitric oxide, which is a very interesting molecule. It's a gas that you can inhale. Your cells make it. It has anti-inflammatory properties, but it also has some antimicrobial properties. And some people have started looking at this as to whether or not this might be a benefit. There are also some molecules or compounds that you can actually swallow that are precursors to nitric oxide. So nitric oxide is a very simple molecule. It's just nitrogen and oxygen bound together. Okay, so before we go on to the next part of the therapies, let's talk about getting to the root of the problem. So all these things that are there that we've done for years and years, all are kind of out at the edges of the problem. They're just working on the downstream result of having this genetic abnormality. And what we really need to do is we need to do something to increase or to normalize the function of the cystic fibrosis channel. So how do you do that? We know that in normal or people who don't have CF, you can see these channels. You get a lot of channels at the cell surface, and you see chloride coming out. And that's the way it's supposed to look. But what you have in CF is you have one of two problems. You either have a deficient quantity of the channels, or you have deficient function. And although many of these abnormal proteins actually do more than one thing badly. So as an example, if we look at little or no CFTR, so that mainly includes the class 1 and class 2 gene mutations. So class 1 are the mutations that have an abnormality where there's a stop sign in the gene, and what happens is you really don't get any useful protein. So you might make a messenger RNA, but you only get a part of a protein. It has no function. It gets degraded. It goes nowhere. Or in the class 2, you have a different kind of an abnormality. So the most common would be the F508del or the delta F508. We've called it for many years. We keep changing the name. So now it's F508del. It's a misfolded protein that gets degraded in the cell and never makes it to the cell membrane. So, you know, the net result is it's as if the gene were almost absent, or it's very similar in causing severe disease as the class 1 mutation. So you might get a few channels, but they don't function very well, and there's just not very many of them. And then you have some abnormalities, we call them class 5 and class 6, where you have some CFTR, and these tend to actually be mild forms of CF. And the class 5 mutation is the one where there's decreased production, so you're just not making it fast enough. And the class 6 are the ones where it turns over too fast in the cell membrane. So you're making quite a bit, but it doesn't stay there very long. It gets degraded, goes into the membrane, might do a little something, and then it disappears very quickly. So each one of these different types of mutations gives you a different opportunity for fixing the problem. So then under function, we have class 3, which is the gating defects. So in this particular family, there are actually 9 of these that we know about. And the most common one's called G551D. It's 4.4% of all CF mutations. And the other 8 gating mutations form another 1% of CF mutations. And what you have in this class is generally you make lots of protein, it's in the cell membrane, but every pore, every CF pore, every channel has a gate on the inside of the cell that opens and closes. And in this type of mutation, then the protein is there, it's ready to go to work, but it just stays closed. And again, it will give you a severe, result in a severe CF phenotype. And then you have the class 4, which tend to be milder, and in this case, the gate opens, the protein is there, the channel is there, the gate opens, but there's an abnormality in conduction, so the chloride and the bicarbonate doesn't move through normally, and that might be because the channel's a little skinny, or there are charges in it that are arranged improperly so that the ions, negative ions, tend to get stuck there, or something, some combination of those things that affect conduction of the chloride ions through there. Okay, so if we look at the 10 most common CF mutations, you will see examples of everything. So the F508del, 83.7% of patients more or less have at least one copy of this. This number's been changing. We used to say it was close to 90%, but there are 2 things that have made this go down. One is that we're diagnosing more mild CF because of genetic testing. So we see these milder forms, and we understand that it's a problem with the CF gene and the CF channel, and people aren't as sick, and we now know they belong to this class of problems. And the other thing is we're diagnosing people earlier because of newborn screening. And the one other factor is that they're also changing demographics in the US. So in some cases this number has gone down a little bit because, for example, in Hispanic CF patients, they have much lower percentage of the 508 mutation than you will find in people like my ancestors who came from Northern Europe, English, Danish. And if you go to Copenhagen, it's about 1 out of every 17 people carry a CF gene. And so that's pretty common. And 80% of those mutations are the 508 mutation. So we assume that particular mutation arose somewhere in Northern Europe and probably in Scandinavia many thousands of years ago. And it's become the predominant one in the world. But you'll see things like G542X. That's one of those, the type 1 mutations that you get this premature stop codon, a message that just tells the cell to stop translating, and so you don't get a full-length protein. G551D, that's the most numerous, the most common of the type 3 gating mutations. R117H, which is one of those mutations that has to do with abundance. And you take the R117H mutation and you combine it with another part of the gene called the poly-T regulatory tract, and if you have 7 Ts or 5 Ts in a row in that tract, you end up with what is essentially a classic CF gene. And 1303K is a missense mutation where you've substituted one molecule for another, and you end up with something that gives you a very severe mutation. And then you have things like the last one on the 3849 plus 10 ABC for T is a very interesting one, in that it's the 10th most common, 1.5% of CF mutations in North America, and it's a partial function mutation. And there are patients, we have one adult in our program who is homozygous, who has 2 copies of this mutation, and who has chronic lung disease, but her sweat test is completely normal, 16. So we still have a lot to learn about CF. It's not all revealed by the sweat test. Okay, so we've learned more and more about the basic defect, and if we really want to change things, we have to get close to that. So, in 1989, when the gene was isolated and sequenced, Dr. Bell enthusiastically and optimistically stated at our Saturday dinner that within 3 years, gene therapy will be routine in CF. So that would have been 1992, and we all know that that didn't happen. It turned out to be a lot harder than anyone knew at that time, but it's kind of a reflection of how naive we all were about those things back in the early days. But you'd like to substitute the gene, and recently people have developed these techniques called gene editing, and they have names like CRISPR and TALENs and Cas, and you're actually able to go in and select a defective gene and replace it with something that works. And it's not been tried in people at this point, but it's a technique that people are very enthused about, not only in relation to CF, but severe immune deficiencies and any severe disease you can think of that's genetically based, which is most of our health problems in humans, a way of fixing genes and making things work properly. And then there's an interesting approach repairing RNA that we'll talk about a little bit. And then the other thing is bypassing premature stop codons, that class 1, all the mutations that end in X. And it turns out that tobramycin actually partially bypasses those stop codons. Unfortunately, it has a lot of toxicity at the concentrations that you have to use to make that happen. But it's the basis for trying to find molecules that will do that are non-toxic. CFTR protein processing, channel function, channel half-life, and then doing something to compensate, going around the defect, finding something else that you can make a substitute for the CF channel. So these are things that people are looking at, and actually it's where we'll spend a lot of the time today. It's talking about CFTR correctors, and these are drugs then that improve the function of the CF protein, but there has to be protein there. CFTR potentiators, these are drugs that increase the amount of CFTR that gets to the surface, and now people are talking about CFTR amplifiers, which is a slight variation on the potentiator. And then there are the CFTR stabilizers, so those mutations that go into the cell membrane and don't stay there very long, or even if they're there a normal amount of time, and you can prolong the amount of time they stay there, and you can turn them on, then that will have a therapeutic benefit as well. So then the other things that we look at is the epithelial sodium channel, which is called ENaC, because it's a lot easier to say than all those big words. If we can do something to block ENaC, which drives a lot of the hyperabsorption in the lung, then you could have a therapeutic benefit. We have lots of drugs that do that. Unfortunately, the ones we tried 10, 15 years ago didn't stick around long enough to do a lot of good. So now there are molecules that are being developed that will block the ENaC channel, that will stay in the body longer and provide more benefit. And then the other thing is looking for alternative chloride channels, because there are other channels that transport chloride. Of course, they may not transport bicarbonate as well, they may not do other things as well, they may not interact with this novel ATPase that's been discovered this year. So how productive that approach will be, we don't know. People have been trying for years using a variety of molecules to stimulate these other channels. And there has not been a lot of progress there. So then once we find something that works in a cell, or maybe in an animal model, and we go to humans, then, classic randomized placebo-controlled trials. So that's what most of these pharmaceutical trials are about. And these are the ones that show that a molecule actually works. Okay, it doesn't say how well it works, but it'll tell you that you can get a statistically significant difference between the drug and a placebo treatment. And so you do this in a double-blinded fashion, so the patient doesn't know what they're getting, the doctor doesn't know what they're giving, and you measure things like lung function or how often people go to the hospital or a number of things and try to figure out if something is really working. One of the big problems they've had is find sensitive, rapidly responding outcome measures. The one that's been around the longest and that we all have access to is the sweat chloride. There's been some controversy about how good that is. My personal bias is it's not perfect, but it has significant utility, and we all know how to do it. We also have long-term clinical outcomes. So when you do, these randomized controlled trials, oftentimes you'll see them go into phase 4, where it's open label trials, and everybody knows they're getting drug, and you look at the lung function and weight and et cetera, and see whether or not the people who are getting the placebo all of a sudden get better and join the other people, and if those effects are sustained. And then one that I think is going to become increasingly important, that you started to hear about in the CF community, N-of-1 trials. So N-of-1 trials are trials where you use the patient as their own control, and you try to figure out in an individual patient whether or not something is going to work. And this is important in part because we've got a couple of thousand CF mutations that have been identified. About half of those are probably disease-causing. And if you look at 1,000 by 1,000 matrix, we know that the frequency of the different genes is highly different. But you've got literally millions of or a million combinations of different mutations that potentially cause disease. And we can't do 1 million controlled trials because there isn't enough time in this life to do that. And in some cases, you don't have enough patients to do a clinically significant classical randomized placebo-controlled trial. For example, I have one little girl who has a novel mutation, pancreatic sufficient, really bad lung disease. How am I going to study her mutation? She's an N-of-1, literally. So the idea that you do studies in individual patients to try to figure out what works is important for that reason. The other reason that it's important, I think, is that when you look at any drug, either a blood pressure drug, Lipitor, and certainly what we've seen in the studies of the CF drugs that we have, these correctors and potentiators, that when you look at the human trials, there's huge variation in response among humans who supposedly are genetically homogeneous. So, they all have 2 copies of 508, or they have G551D and 508 in the original ivacaftor studies. And you look at the change in sweat chloride, for example, or the improvement in lung function, or whatever measure you look at, and you'll see huge variation in clinical response. So I think I've become convinced that we have to treat every patient that we take care of as an N-of-1 trial. If I'm ever going to figure out how to best treat a particular patient, I better look at things very carefully and figure out what works best for them. And that's going to become an increasingly important aspect of how we care for CF, I think. Okay, so here we are, we're away from the edges, we're away from the downstream. Now we're trying to fix something in the cell itself. And so we have 2 drugs, ivacaftor and the ivacaftor-lumacaftor combination, that are now actually in patients. Ivacaftor we'll talk about a little bit in people with gating mutations. It's set a very high bar for anything that comes afterwards because it's so incredibly effective that people's lives have been transformed by this drug. And then there are other patients where it doesn't seem to work quite as well, but most people who have these mutations have benefited greatly from this drug. Lumacaftor, ivacaftor, or Orkambi, which is the brand name, is less impressive. And we think that people recognize this because we know that there are things coming after it very quickly. For example, if we look right here, VX661 and ivacaftor, this is now in phase 3, studies this particular drug combination. VX661 is the next generation or the next iteration of the glumacaftor. And it actually has a name now. It's called tezacaftor. I just learned here recently. It has a name. It doesn't have a brand yet, but it has a name, tezacaftor. So tezacaftor, ivacaftor combination is in phase 3 trials. There's hope that it will have some advantages over lumacaftor. There's hope that it will be more effective, but we really won't know until we actually see the data. And then CPT-656, this is an interesting product that actually the company took ivacaftor and they put deuterium where there's hydrogen, so it's deuterated. It's not deteriorated, it's deuterated. Okay, it's a deuterated molecule, which then makes it last longer. So then you're talking about once a day dosing, which could help drive down costs, improve adherence. It may have some other advantages. So that's an interesting molecule. And edalurin is one of those. It's a molecule that's intended to overcome the type 1 defect. In the first trial, they didn't meet their endpoints. When they did their sub-study analysis, it looked like people who were taking inhaled tobramycin, there was an inhibitory effect. So now there's another phase 3 trial going on, going back to see if you don't do the inhaled tobramycin, do you get a better benefit from edalurin? So this is the one that promotes read-through of that premature stop sign. And then there are some other drugs in development. This is not an exhaustive list. These others are molecules that are looking at alternative chloride channels, anti-inflammatory, or other things that have some benefit. So those are all in phase 2. So phase 3, and what we'll hear about perhaps later this month and next year will be the studies in edalurin and VX661 ivacaftor combination. And then the list goes on. You can see how many there are here. I couldn't get it all on one slide, so we've got all these other molecules, and I won't spend a lot of time talking about these because these down here, actually they have grants from the foundation to develop certain kinds of molecules, but they're not even in phase 1 yet. They're in the laboratory. They're working with cultured cells. They may work with animal models at some point, but very early in development. And then these others are slowly moving on. So let's talk about the first one that actually made it to patients, which is ivacaftor. So here's a diagram of those channels, and so they're in purple. And you can see that they're there, and a little chloride gets through, but not very much, because this is a mutation that results in a very severe phenotype. And if you look at these patients, 508, 551D patients, average sweat chloride is about 100, which is classic CF. They're pancreatic insufficient, so they have to take enzymes. They're not in that small group of patients who don't need enzymes, which is a sign of function. They have all the signs and signatures. Their disease progresses rapidly. They get very sick. In fact, until this drug was available, the sickest patient in our center was somebody who had one of these gating mutations, and she was only 12. Okay, so the drug opens the channel, and they do these things called single channel studies where you take a micropipette with a really small tip on it, and you pick up a little piece of membrane, and if you get the right piece, then you can see one of these channels. You can actually measure ions going through it. You see these little microcurrents, and you expose it to various things to prove what it is, and you vary the voltage across the piece of membrane to do what's called a current voltage plot, and you can demonstrate that it's a CF channel. It has certain characteristics, and you can see the channel open and close. And so you see these time plots where they open, close, open, close. So it's, they're either on or off, and not ever in between. And you can see the change when you expose the G551D or any of the other gating mutation channels, you expose it to this drug, and you see a very dramatic change in how those things function. Oops. All right. Now what did I do? Okay, so let's go back and start this. I'm
It's okay.
sorry. On the current slide, here we go. Oh. That's not the current slide. Okay, we'll just go backwards real quick here. There's a key right next to that key that makes you go right to the end. I didn't know that. Okay, there we are. All right. Okay, so how does this improve things? Well, you see a normal chloride channel over on the left here, where the little green chloride ions are floating through there, and you've got a pore. There's lots of channel around. When you have a gating channel, you can see you have lots of channels around, but they're all shut, and there's no chloride coming out. If you can open them, which happens occasionally without treatment, you do see that the conductance is actually pretty normal in these channels. But the other thing is you've got a deficient quantity. So your total chloride transport is going to be a product of how often they're open, what the conductance is like, and the number that you have at the cell surface. And so ivacaftor does a great job of this. I'll show you a little bit about how much this improves it. It's now approved for the 9 gating mutations. That we used to say there were 10, but the 10th one, which did not respond to this drug, people are now saying it was misclassified and it's not a gating mutation. It probably explains why it didn't respond to ivacaftor. And then recently, this drug was approved for the R117H, which is this milder mutation that causes more severe disease if you have these changes in that regulatory part of the gene called poly-T tract. And recently, new this year, it's now approved down to 2 years of age. I can tell you though, we can talk about experience, and we've now used this in an even younger child who was doing badly without it. You can argue that point with Medi-Cal, CCS, and with your insurance company. Okay, this is the famous Wolchansky plot that Wolchansky never plotted, but a replotting of his data and analysis shows that what we're trying to do is we're trying to get down here where normals and carriers, if you look at their sweat chloride, they're down somewhere in this range, certainly under 30. The average in normals, not carriers, is somewhere around 16 millimolar. You have these things called CFTR-related diseases. These are CF mutations that don't cause lung disease, but you might get sinus disease, you might get polyps, you might have problems having babies. It affects the male reproductive tract, and that's the only abnormality in general. We've had a number of patients come to us from the fertility clinic. But then it's up in here where the business really is, where the classics are up here around 100. And you can see this part of the curve if you plot it out and you look at sweat chloride versus activity of CFTR. It has a fairly steep slope. So if you can start moving the sweat chloride down this slope, you start to improve your activity. So you go from something close to 0 to 20, 30, 40% of normal. And the experience that we've had with ivacaftor and gating mutations have shown that that was absolutely right. Before the ivacaftor studies were done, I used to kind of, on the sleepless nights, I'd lie there and think, well, what if we fix this mutation or this protein and nothing good happened? What then? And I imagine other people had the same kind of, I don't know if that's a morbid thought or a dark thought, but it was a discouraging thought. But it turned out that I was being overly worried, overly anxious about it, and it turned out that, that when you looked at the gating mutations, the G551D data, that if you look at chloride transport in these patients, 508, 551D, almost no transport at all under baseline conditions, and Here you get up to close to half of normal, which is an incredible change. That's like transport you would see in a carrier, right? It's close to it. Carriers we know are fine. They walk around all the time, and there's no scarlet letter anywhere on their body advertising that they're carriers. They look just like everybody else. I could be a carrier. I've never been tested. Father's ancestors certainly came from the right part of the world. Chloride transport in CF patients then, or change in sweat chloride, the average change was close to 50 millimolar. So, it dropped a lot. In the placebo group, there was, of course, no change. And when they looked at change in lung function, there was a robust change in lung function, but it wasn't huge. Part of the reason for that is that this population was a very diverse population, and there were some of the patients in these studies actually were pretty healthy, and they couldn't improve that much, and there were probably some that were so sick that they weren't going to get much return of function. But then there were some, the majority of them had some improvement. And when you look at the data, the way it was presented, you see these sustained improvements, very significant improvements in FEV1. The first exacerbation separated very quickly. Everybody gained weight. In fact, some of them gained too much weight, gained 10 pounds. And we're always working to keep everybody fat, and they're on these high fat, high protein diets. And you fix their abnormality, all of a sudden, they're turning into little chubby kids. So we had to change dietary habits on some of these patients. So I just have here a collection of our own patients, our pediatric patients that are on ivacaftor. One of these has, they're all 551D except for one of them who has S549N. I'll show you a little bit about that. So you can see what their sweat test did, going from 95 to 20, 83 to 24, 88 to 41. The most disappointing one is the one who just moved into our center not too long ago, went from 98 to 58. And one of the things that we played with, we thought, well, maybe he just needs a bigger dose. Didn't make any difference. We checked on his adherence. He seems to be taking things as far as we can tell, and he just doesn't respond. And then this 100 went down to 32. Let me talk about him. He's our resident flake. He can't even take a pill twice a day to save his life. And he actually, his baseline was 100. And when we had him come into clinic, he was like 60. And he admitted not to being very faithful. And we ended up, he's the only one of all these. He did come in, we put him in the hospital because he was losing lung function. We put him through an IV antibiotic course, and we made sure that he took his ivacaftor twice a day, and he went down to 32. So there is dose dependence, but if you don't get that big response, there's still individual variation here, and you can see, there are patients here who actually get down into very normal numbers, and then there's some where 41 and 32 is still a little on the high side, and the 58 certainly is high, and we tried as best we can to make sure that they're all taking their medications.
Time frame for that?
For the change?
Yeah.
You see most of the change in sweat chloride within about 3 days. But certainly by the end of the week, you're in a steady state. So it doesn't take very long. You just have to take all your drugs for a week, and you're there. And most of the trials, they did 2-week points. Although in the very first trials, first phase 2 trials, they did sweat chlorides every day, and you can just see how they march up. Okay, so follow up on ivacaftor. Those patients, there's only one who's been admitted, and that's Mr. Flaky guy who can't take his pill twice a day. But they don't cough, they don't produce sputum. The sickest one had an FEV1 of 33% of predicted when we started. This is our S549, and she's a case report. And she was, we figured, and she was sick. She spent so much time in the hospital. She hadn't been home for Christmas for 5 years. She's a 12-year-old. And she's a great patient and a great family, and it was just bad disease. We put her on this, and the S549N mutation in cultured cells, actually, there's a 100-fold increase in gating. and you actually go up to normal levels of gating, normal probability with this particular mutation in the cells that Van Goor studied years ago. So this is a genotype that's very sensitive. So she went up in 4 or 5 weeks. She was up 50% of predicted. She stopped coughing, stopped producing sputum, and her X-ray changed, her CT scan changed by 6 months. I scoped her once to just see what was there because we couldn't get any cultures anymore, and her airways looked completely normal. She still grew out a little bit of the bugs that she had, MRSA and a stenotrophomonas, but she's not been in the hospital, and she spent 116 days in the hospital over the 2 years prior to starting. And the surprising thing was that we continued to see improvement in FEV1 in the second year and even in the third year. So she's gotten as high as 78. She had a little downturn last week, so she's going to get a little treatment. This will be her first exacerbation around 3 years. So, this was a miracle. 2 more of those, and the Pope would have to give me a crown. Nobody's had IV antibiotics since starting except that one patient. And I think that, looking at these really sick kids, some of these 551D kids were actually pretty healthy. So they didn't come in the hospital, they maintained, but the very sickest kid is the one who showed us the real power of this approach, that pharmacologic manipulation of this gene and this protein can do incredible things. So, gene editing someday will be the treatment of choice, but I have no idea how long that's going to take. And so if you look at chloride transport percent of normal, so normal baseline is 100%. Ivacaftor actually stimulates normal and drives it up by another 50%, so more open time even with normal CFTR. 551D goes from 1% of normal to 55 with, this is 10 micromolar. Ivacaftor, I think, is the concentration. S549N, our little girl that was so sick, it goes from 2% to basically 100% of normal with this drug in its environment. And those responses clearly, in our experience, have translated into clinical benefit. So what we need is drugs for everybody. Ivacaftor, though, does have use in more than just the patients with gating mutations. Those are the ones that have the most dramatic response. But this group, the R117H patients, have also been looked at. And you can see that if you take an R117H, I think this was R117H7T, went from 33% of normal to 134% of normal. So if you have 508, R117H, you can potentially get up to something like a carrier, or actually a little better than most carriers, just based on those numbers. And we've seen that happen in our patients. So in this study, the drop in sweat chloride was not as dramatic. It was about 24 millimolar, so not a huge drop. But what they did see, oh shoot, what is that button? I keep hitting it. It's right next to the arrow. I'm sorry. Okay, back to slideshow, current slide. And we'll go back up again. So there was the sweat chloride change. Here was the change that they saw in lung function. You can see these changes aren't huge. Statistically, they barely meet criteria at the end of the study period, so that was 24 weeks of the drug. So not huge changes in lung function. And maybe a little weight gain. So certainly much less remarkable than the people who have these gating mutations that are very sensitive to this drug. But there's certainly a signal. And what I can tell you is that we have tried this drug in a number of patients. We called it the same study, short-term ivacaftor and non-gating mutation patients. And we had a patient who had 508 and this mutation, which is a partial function mutation. And he's been pancreatic sufficient his whole life. He's, was healthy for years and years, but now he's 60. Okay, and if you have mild CF and you're 60, stuff has happened. And his FEV1 is down around 50% of predicted. His chest X-ray is abnormal. He coughs up sputum. He has chronic sinusitis. He can't gain any weight. We, under the auspices of our privately funded trial, put him on the ivacaftor. And what we saw was, and this was blinded crossover, placebo for 2 weeks, ivacaftor for 2 weeks, only a 2-week trial. And what we saw was a greater than 20-point drop in his sweat chloride, and we do repeated sweat chlorides so that we can get a mean and standard deviation and standard error and compare our baseline with our on drug. And then, we did sweats at the beginning and end of the placebo period and the study period, the drug period. And we didn't know which was which. And initially, with 2 weeks, he couldn't tell any difference. But he had enough of a change that we went to his insurance company and we said, we think this guy would benefit from long-term ivacaftor, and we want it. And we wrote the prescription, and we arm wrestled a bit, and we got it for him. And he's now out a couple of years on ivacaftor. He's gained 10 pounds. See, I thought I put that slide in here somewhere. Yeah, here he is. Okay, so 2 hospitalizations per year. He feels sick. Oh, he's a G542X. I'm sorry, it wasn't 508, but that wouldn't matter because it doesn't work on that one either. Just the 3849. And this is what happened. The sweat chloride went down from there to there. His FEV1, the top number is actually his weight. So he was less than 70 kilos. And, out, let's see, that was May. So that was less than a year here where we plotted this, but, he's gained 10 pounds since he's been on it. And his lung function, not very dramatic, but it didn't go down. And he hasn't been in the hospital. And he doesn't cough anymore. And his sinus disease is better. And he calls it his little health pill. Okay, so is that worth the cost? That's a socioeconomic argument. I don't have any way of answering myself, but certainly for him, this has made his life much better. And now he's a 62-year-old with mild CF who really acts like he has mild CF rather than gradually deteriorating and having his life shortened by, 10, 15 years. So hopefully he'll live out his otherwise normal lifespan and not die of CF, maybe die of old age, like all of us hope we will someday. So, lumacaftor, ivacaftor combination approved, tezacaftor, ivacaftor in phase 3 trials. I'm assuming if this drug looks good, it'll get submitted to FDA. We're all hoping that it will look good. One of the things that you didn't see on that chart and that you won't see when you go to the pipeline is that there is another step here, and that's the triple combination. And so, there is a triple combination that has gone through phase 1 trials that includes tezacaftor, ivacaftor, and a third drug. There is a company that has not been in the CF game before, who is working very hard to, they're now doing, they have a corrector, 2 correctors or more, and 1 potentiator or more. And they're doing monotherapy, doing the toxicity trials, and they're going to go to a triple combination for their clinical trials. So they're going to try to get to that point very quickly. And this actually makes sense. When you look at the pathophysiology, my bench basic research friends have all looked at this, and for several years now they've been saying, you need more than 1 corrector. It's just not enough. You've got to correct this thing in at least 2 places in its metabolic pathway in order to get it in good shape into the cell membrane and then turn it on. And we know that ivacaftor actually is pretty it's not very choosy about which CFTR it works on. If you look at the studies for temperature-corrected 508 cells, different cells, it will potentiate almost every mutation that it's been used against. So it has a pretty broad spectrum of action in terms of the mutations. And the problem really is in the corrector part where you're trying to actually increase the amount of protein that gets through the cell and gets put into the membrane. So triple combination is, I think, pretty clearly where we're headed. And lumacaftor, ivacaftor, I suspect will have a short life in our armamentarium and will be replaced by tezacaftor, ivacaftor with something else that has a number for a name. And there are phase 2 trials that are getting started late this year and early next year. And by a year from now, we should have enough data to say whether or not that combination is going to move into phase 3. So that's moving fairly quickly. And hopefully it passes muster and does a great job. And there are reasons to be optimistic. There are cell studies that suggest that that triple combination has the potential for being very effective. But the potential for being effective and actually being effective are not the same thing. And we have to do the studies and do the measurements to prove that this is really going to be a step forward. But this may be where we get to something that I would feel comfortable calling a breakthrough for 508. So these are some of the summary of data from the cell studies. So here's lumacaftor, ivacaftor, and the first one was in 508 cells with G542X, and you can see when you put both drugs there, there's a bump up in activity, presumably not related to G542X because the drugs that are in this study are not really targeted on that. And you can see that when you do 508 cell and you put the two drugs together, which is the one that's in the open box, you can see that the bump up is even bigger. It's about twice as big as it was in that set of experiments with the 508 G542 cells. And that's what you'd expect, because that's one 508 to work on, and that's two 508s, and so you get twice as much effect. And you end up seeing twice as much chloride transport. And then 508 G551D, so that's the gating mutation that's most common and very sensitive to ivacaftor, and you see you put that combination. When you do just ivacaftor, you're seeing about half of normal chloride transport. You add the lumacaftor, and you get something that's over 80% of normal. So the combination actually, there is some synergy there, and there's a suggestion that our gating mutation patients who have 508 at least, may have some benefit from using combination therapies. But we're not yet at the point where we have enough human data to make that recommendation. That has to be proved in clinical trials. So here's the triple combination, so lumacaftor, ivacaftor, and the second corrector. And this is what gets really exciting, because you see here, if you look over on the right, the 508 cell line, and you have all three drugs, you're actually up around 50% of normal. And the tezacaftor with ivacaftor and the second corrector have something very similar. In fact, in the cells, it's a bit stronger than that. So, you see something that really looks promising. And if you look at the 508 G542X, so this really only has one gene to work on, you see something that is lower than the 508 cells, but you still see a significant benefit. So this is exciting because the Orkambi lumacaftor ivacaftor combination showed no benefit in people who only had one 508 mutation. That was disappointing. But what this says is that if you take and add the second corrector and maybe improve the first corrector, you have the potential for really making a breakthrough in chloride transport, which again should result in significant clinical benefit. So there you are. There's the master plan for this. when we get through all these randomized placebo-controlled trials and we have a bunch of these correctors and potentiators around, I'm convinced we're still gonna have to come back to the N-of-1 trials, because I have patients who have these really odd mutations. Homozygous for Q890X, parents are third cousins, kid's pretty sick, or he's a young man now. We have others like that have two unusual, uncommon mutations. And we've targeted 508, and that's going to help a very large percentage, but there's still, and the number keeps increasing. You're talking close to 20% of CF patients now don't have a copy of 508. It's low 80% that have one copy, so it's 17 to 18% currently don't have a single 508. So we have to use these drugs to see if they work in other mutations, in other settings, and in various combinations. And we may need to dose them differently. One of our patients that we've tried ivacaftor in has a novel mutation. It's the only one that's ever been described in the literature. She's pancreatic sufficient, but right after she was diagnosed, she came down with a severe infection of her lungs, and her lungs were trashed. So she now has severe CF lung disease, still pancreatic insufficient. And we tried her on the ivacaftor fairly early on, and she was small, she was young. We didn't know a lot about dosing, so we just gave her the full dose. We didn't have any half doses. And we were told not to cut the pills in half because they couldn't guarantee that. And now we would do this all differently. But we tried her on the full pill twice a day. And her sweat chloride came down 31 points. She went from 60 to 29. And we didn't have her on it long enough to see if it did any good because her liver function started skyrocketing. So we had to back off, and we know that you see that. So, we waited a couple of years. She continued to struggle a little bit. She's her grandmother is her guardian. We sat grandma down one day and said, she's coming in the hospital. She's not gaining weight. We'd like to try it again, but we'd like to try half a dose and see what happens. So now she's bigger, she's older. We put her on half a dose, because now we have half doses available to us. We saw the same 31-point drop in her sweat chloride, and her liver function's been absolutely stable. And so we were able to find a dose, luckily, that gave her apparently all the benefit without the liver toxicity, or the presumed liver injury that at doses that these drugs, ivacaftor in particular, causes occasionally. She's been great. She's not been in the hospital since we put her on it. She doesn't cough anymore. All those things that you want the drugs to do, it's done for her. She's gained some weight. She's a volleyball and softball player, and so she's very athletic, and she's out there running around and having a good time with her life. She doesn't meet any of the criteria. So people have asked me sometimes, well, how do you get this for your patients who don't fit the criteria because nobody wants to pay for it? The first time we did it, we were lucky because CCS had never had a prescription for the drug before for someone who wasn't covered by the guidelines at that time, so they did it. We saw this wonderful response. The next time we tried to do it, they said, whoa, wait a minute, this is going to break our budget, why should we do this? But what we have done is that we have looked very carefully at the sweat chlorides before and after being on, in this case, ivacaftor. In a few years it'll be various combinations, I think. We showed that there was a statistically significant drop in the sweat chloride, and then we followed them along carefully to show that improvement is maintained and that something else happens. Like, in CF studies these days, our most sensitive indicator of effect is frequency of exacerbation. So what I can tell you is, in all these diverse patients that we put off-label onto ivacaftor because we saw some change, like mainly a drop in sweat chloride, significant drop in sweat chloride. Not huge, but significant. What I can tell you is, except for that one, Mr. Flaky Boy, nobody's come in the hospital. Nobody's had an exacerbation. or at least not a serious one. That's a meaningful endpoint, and it's a very diverse population, so it's hard, thinking about how to write up this paper, but it's a series of N-of-1 experiments, and probably the most sensitive objective indicator is that nobody's needed IV antibiotics. And we've been doing this now for a number of years. So we're convinced that this is a good thing, and I'm convinced there are other people out there that benefit from ivacaftor by itself. When we see these other new combinations come in, we're going to want to look at that triple combination or the one that comes from the other company that's a triple combination. And we may find that people respond differently to, there may be somebody who responds really well to lumacaftor. It's just not the 508 patients. You don't see the kind of massive correction and throughput of protein that you want to see with that particular single drug. The triple combination gets closer to that, but there may be other people out there who are much more sensitive to the effects of the lumacaftor. And I would argue that we need to keep open minds, and we need to have the freedom to do these N-of-1 trials, but then we have the obligation to prove that these things are really working, because these drugs, A, have potential for doing harm as well as good, and they certainly do harm to whatever our health budget is in this country. I mean, these are expensive drugs, and that's the other harm that we do, is that we don't want to bankrupt CCS and Medicaid. They're our allies most of the time. Sometimes we arm wrestle, but we're on the same team most of the time. We're there to do the same thing, which is to help people. Insurance companies, we want to be able to work with them, continue to work with them, and show that there's benefit. And so I think that we need to, cast a wider net in part, but then we need to carefully document what happens when we do that. We can't just, here, take it. See, tell me how you feel next week. That's not going to work. And you can get into arguments about how good sweat chloride is, but I also know from having talked to a senior scientist at Vertex that, I was complaining to him that I couldn't measure serum volumes. Oh no, serum volume, I want to do pharmacokinetics on each of my patients so I can understand whether that's the reason some of them don't respond as well. Is their pharmacokinetics different? Do I need to run the dose up? Do I need to change the interval? What do I need to do to make this work better? And he looked at me and he said, just measure the sweat chloride. That'll tell you more than the pharmacokinetics will. So he didn't elaborate, but I think that that's telling me that having the drug levels is not going to be the answer. I'm looking for a sweat chloride response, or until somebody has something better than that, but we all can do sweat chlorides. And then looking longer term at the clinical response, changes in cough, changes in exacerbation rates. That's really going to answer the questions for us, but it needs to be very carefully documented in each patient's charts, so that if somebody comes back to us and says, hey, we don't want to pay for this expensive drug anymore, why should we do that? Then you have data. I can tell you that when you talk to insurance companies, sometimes you get referred to the clinician, their consultant, and so far in my conversations with consultants, if I have data, they have no argument to counter that. And I now have experience where we've taken this drug and used it in places where the company can't say, and I've asked Vertex, well, why don't you give me a starter pack? And they say, oh, no, we can't do that because regulations prohibit them from encouraging off-label use. There are federal laws that would put them at risk, so they can't do that. I don't, I would change the law if I could, but it's probably not going to change. So we have to be creative in how we do these things. And so far we've been reasonably successful at getting this drug for people where we think there might be a significant response. And then, again, just documenting how they respond. And if they don't respond, then you should stop. Any questions?
And if you have a question, we have the mic because we are recording.
I like this picture. Oh, I love the friends.
I'll ask a question.
The mic doesn't work.
The mic does not work?
No.
Oh, so sound doesn't matter where I...
You're
We're
speaking
using
of
the
the
mic on
camera.
camera.
Yeah,
Oh, so I should
mentioned
stand
this
near
at
the
beginning.
camera.
Ivacaftor is approved down to 2 years of age, but we had a baby that was a year old that we started on because she had a very bad winter, 4 hospitalizations, really sick. And, you know, we argued that when we won. We got her on. It took a little arguing, but we got it for her outside the guidelines. And we guessed at a dose. We followed her liver function. We followed her sweat chloride. And she's done beautifully on it. And she's a G551D508 patient, so classic gating mutation. So we knew she would respond. The biggest challenge was, you know, getting the dose right so that her liver function didn't go up. And she had a nice response on her sweat test as well.
So my question, well, I have a daughter who is double Delta F508, so I'm waiting for all those new therapies to come down the pike. But one thing I had heard, and I can't remember where it was, if it was at a conference, about, and this comes to the whole isolation issue with cross-infection, that especially with ivacaftor, that there were people who had mucoid pseudomonas, had been colonized with it for years, who after being on ivacaftor were able to actually clear for the first time. Have you heard of that?
Yes, and you know, I mentioned our one patient who had a different gating mutation that we put on ivacaftor, and the only way that we got any cultures from her was to do a bronchoscopy, because otherwise she didn't produce any sputum. She was so healthy, and that was 6 months into treatment, and everything looked normal. There were a lot fewer white cells in her sputum, and normally when you look down a CF lung with a bronchoscope, you see what I call the river of pus. And you know, people are lying on their back, and you just see this stream coming up from both sides, and it's really kind of ugly. And then the mucosa looks patchy and red, and if you touch it, and it starts to bleed a little bit. And she didn't have any of that. It was all gone. There was no pus. Secretions were actually quite thin. The other thing that you see that I don't think it's actually reported anywhere, but if you go in and you do bronchoscopies, which we don't, you know, have done rarely, but a few times I have, you do it in somebody with CF who's young and healthy, you may not see the pus, but the secretions are still kind of streaming, and that's because the composition is different than normal. And in her lungs, you know, I went to suction and get a sample for culture. It was just like somebody that didn't have CF, and we haven't re-scoped her because she's done so well. So I don't know if a little bit of MRSA and steno that she had at 6 months is still there. It could be gone, but she doesn't produce sputum, so it's hard to culture. You know, we did throat cultures, and those aren't really very good, especially in this circumstance. Sometime we may have an opportunity to go down again and sample and see if those are gone. But yes, I can't say that we've seen that, but we haven't gone doing bronchoscopies in all these kids to see if their bacteria have gone away.
And is that anything just nationally that's being tracked in CF centers? Do you know? I mean, is anybody looking to see?
I don't know that there's, currently there's not a good way of tracking in the registry that I know of. And the other thing that's happened is some people have seen their CF-related diabetes get better. So that's also happened. So presumably that's related to stress in the pancreas on the islet cells. So when you take the stress off, they work better. They still don't have pancreatic ducts, and it doesn't usually correct pancreatic insufficiency. And I've looked a couple of times. I've just gotten some poop from some of our kids with gating mutations who are on drug doing well, and measured the elastase levels in their stool, and they're still very, very low. So they're still pancreatic insufficient. But apparently there's enough effect that some people have seen benefit for the CF-related diabetes part of CF.
I have 2 online questions.
I guess you have to stand with the camera.
Oh, okay.
So somebody stuck with us?
People post it on Facebook too.
Oh, okay.
With more companies entering the field of CFTR modulation, do you think this will lead to competition that might lower the cost of the medications, or will they all be expensive?
Yes. It will probably lower the price, but they will all still be expensive. You know, if you
Yes,
take,
to the second degree.
you know, $300,000 a year and you cut it in half, it's still a lot of money that most people can't afford. I don't think there's a single solution to that problem. Competition helps. Myself, I'm not a believer in trickle-down economics, and I don't think that competition works very well in medicine because it never has. I think there are other mechanisms that have to be put in place, like you have to allow Medicare and Medicaid to negotiate prices with drug companies, like they do in Europe and Canada, where they're able to drive the cost down to the patient by negotiating with the manufacturers. But in this country, the manufacturers have free license to charge whatever they feel or whatever they want. You know, we've seen that with the EpiPen recently and with some other drugs that have been in the news. And I think that these drugs sort of fall into that class. If you ask the people in the pharmaceutical industry, what they will say is that we're still losing money because these development of these things is so expensive. We not only have not repaid our investors, we actually are still losing money because we're still doing clinical trials and so on. So, you know, they have an argument, and so I don't think competition by itself will do this. We need to figure out ways to decrease the cost of doing clinical trials. We need to stop companies from doing Me Too drugs, maybe. Maybe we need to change patent laws so that generics don't come out for longer periods of time, so companies have more time to get back their investment and stop thinking about, well, we didn't make any money, we lost money this year. You know, a company that survives 100 years only has to have a positive bottom line at the end of the 100 years, not in year 2 or 3 or 10. And these are developing industries where getting to a positive bottom line can potentially take a long time. So, you know, I'm not an expert in this, but I've thought about it, and competition by itself won't do it. Generics by themselves won't do it. Case in point, Zantac, generic Zantac. Unless you go to Costco, it's still about $1 a pill for a generic, I think. Last time I did that, I go to Costco, so it's but you know, that's how much it costs as a prescription drug for a brand name Zantac, and the generics just really didn't drive the price down that much. And I don't think that they have. And if you look at the other CF drugs like Pulmozyme and inhaled tobramycin, when those first came out, they were $11,000 to $12,000 a year, and now they're $25,000 to $30,000 a year. So they're not like computers. The price hasn't come down with time. The price hasn't come down with generic competition. You know, there are other factors that keep those prices really high. So I think that's a very complex problem that we're going to have to find the political will to deal with at some point, and get some people who are much smarter about these things than I am to figure out how to finance this.
Thank you. The next question is, what about treatment time? People talk about medication development all the time, but I would love more focus on technology that will shorten the amount of time I'm nebulizing medications. Is there anything in the works?
Well, yeah, we showed some of the dry powder inhalers for the inhaled antibiotics, and that's a huge savings, time savings, because the inhaled tobramycin, nebulized tobramycin takes a long time. Casein is quick, but it's 3 times a day. So, you know, you get people preferring one over the other for various reasons. The dry powder inhalers make it quicker. I still have patients who say, I'd rather do the nebulizer solution. I don't like the feeling of that powder in my throat. So, you know, we see that happen. So they don't care about saving time, which is kind of perplexing, but they don't. I guess they're just used to it. Change is always hard, even if it's good change. Yeah, in terms of time savings, you know, one of the really big unanswered questions, now that we are starting to see correctors and potentiators become effective, the question will be like, you know, the baby with 551D that we started on ivacaftor. And even though this baby was in the hospital a lot, her CT scan still, she doesn't have bronchiectasis, okay? So can we prevent the bronchiectasis? will she be able to grow up and not have to do 2 or 3 chest treatments every day? Will she be able to maintain her good health without doing pulmazine or inhaled antibiotics or other things that take time and money? I don't know. I don't know the answer to that. With our patients who have started on these drugs that have had disease for years, we're not backing off any of those things, but they have pre-existing damage that we think makes it unlikely that they can just stop everything and depend on the pill. And we've had some patients who've done that on their own, and what happens is their lung function starts to slip. And then we have to go back and re-enthuse them and treat them and get them back on everything. So we're pretty sure that if you've had CF for 10 or 12 or 20 years or 60 years, it's not going to reverse the damage. But if you have a baby that you start on the drug and you prevent the damage up front, then what? Can you not do some of those other things? And that's an unanswered question, but that's, I think, one of the interesting clinical questions for the next decade or so.
That's all I have for now.
Oops.
Tight quarters. Thank you, Dr. Nielson, so much. That was an incredibly informative presentation, and I actually have a whole list of questions, but I'll ask you later. So thanks to everyone who joined us tonight. Thanks to everyone who's watching this. And I want to give a special thanks again to Vertex Pharmaceuticals, Genentech, and Chiesi USA, who are the sponsors of tonight's program, as well as one thanks to Gilead Sciences and Novartis for your additional very important support. We look forward to seeing everybody next month on Thursday, November 10th, when our speaker will be Dr. Rick Moss from Stanford, and he's presenting on a topic that impacts many people with CF, which is fungal infections in the CF lung. Before we end, I just want to encourage everybody to go to CFRI's website and just keep track of all the programs we're offering, our MBSR classes, our support for individual counseling, and our CF caregivers support group. We truly want to be your partner in living. So thank you all very much for participating, and thanks again to Dr. Nielson. Please have a good night. Thanks.